So welcome to molecular biology. Yes, you're all in the right place. This is not classic Platonic Socratean discourse. No, it's not the right place for that. Hopefully, again, you're all in the right place. Did everybody have a chance to look at the lecture on YouTube from Monday's lecture? Anyone not see that? I know some of you aren't registered for the class yet. So those of you who did, what are your questions? If any, yes. Mm-hmm. Yeah, so the question is, I will repeat this because, again, I'm recording all of this and they'll all be put on YouTube. Question is about the 5th edition versus the 6th edition of the textbook. I've had a lot of people ask me that question. I know the 5th edition is a lot cheaper than the 6th edition. There are some things in the 6th edition which are not in the 5th edition. And as I mentioned right before class, the 5th edition was published in 2008. That means it's data from 2007, which may sound like a relatively short period of time ago, but as far as molecular biology is concerned, that's almost a generation. And this one is from 2014. So particularly things as far as RNA, some of the regulatory aspects of things, and then again, there's been quite a lot of progress in that last period of time. In fact, we'd almost given up on them giving us a new version of this textbook, and then they came out with one. So we're actually really quite happy about it. What we're trying to do to lower costs is there is a paperback version, which is a lot cheaper. There's also the fact we're using this book also for the cell biology course. And so, again, I'm fully aware that textbooks are an issue. High costs, et cetera. We're doing our best to try and deal with that. Unfortunately, the way the field progresses, it's a little bit tough to do that. I'm going to try to get a copy on reserve in the library of this version. So if you have the 5th and you want to pick up on the 6th, that's there. So that's the textbook issue. And yeah, sorry about the post. It's there, but it's dug way, way down. It's hard to find. Any other questions? Yeah. Okay, the Kindle version apparently is a lot cheaper, and it's really not bad. Yeah?
Same with Amazon renting. It'll let you keep it until the end of May.
Okay, so apparently you can rent on Amazon until the end of May, so that's another option. Okay, I personally am a Luddite, so I like the real book, but yeah, I'm old, what can I say? Yeah?
I had another question.
Sure. Yes.
Does that refer to any homolog throughout any level of time, or is it talking about species?
Okay, so the question is about how to define gene families. And correct me if I'm paraphrasing incorrectly here. How to define gene families, how to think about gene families. Basically, the way I like to think about gene families and the way that most people do is they're a product of some kind of gene duplication, either through speciation or through gene duplication in one particular organism. So they can be orthologs or paralogs, and they're all going to be homologs, but homologs by the definition that they're related ancestrally. So they may or may not actually have detectable sequence similarity to each other. And so we'll talk a little bit more about that in the lecture today. Yeah, so the question is, could they be in different species? Yes, very much so. You can have protein families that will be present through lots of different species. And are those going to be orthologs or paralogs if they're in different species? Yeah, they're all going to be, now, if they're in different species, okay, well, I heard ortholog and paralog, and I haven't pulled up that as a clicker question yet. But so, how many think orthologs? How many think paralogs? How many are totally confused? Nobody admits to being confused? Wow, okay. Well, we'll get there. So if you think about orthologs versus paralogs, the way I like to think about it, pair for pair, as if it's formed a pair of genes in one organism. And so a paralog is going to be in the same organism. Orthologs are going to be in separate organisms. So that's the way I like to think about it. That makes sense? Yes? No? Paralogs and orthologs is a really important concept. We'll talk about it quite a bit throughout the rest of the course. So it's an important concept to get down now, early on. Okay, so a couple of other things as far as what I went back and looked at my lecture. And I also noticed that I'm standing in front of the screen down here, which is a mess. It's a problem with this classroom. I'll try and not have too much here down in the corner. Of course, times are different this year. I reserve the right to have in-class clicker questions. Not everything right at the beginning is what I've had before. At most, there'll be 1 or 2 per class session, and probably less than that. But just consider yourself warned. No reef polling. I'm barely getting used to dealing with the whole clicker thing myself, so don't bother with the whole reef polling business. Office hours, of course, are right after class, which is not the same time as they were in the past. Guest lecture on January 15th. I'm going to be out of the country. I found this out way before knew that I was going to be teaching at this particular time. So there'll be either a guest lecture, or we may also do a posted from previous years, et cetera. We'll see how that works. Anybody have a chance to check out the genome exhibit at OMSI before it closed? Very cool, really nice, and actually there's a lot of nice connections to sort of the second half of the lecture on Monday. And then the other thing here, which is important, for registering your clickers, use your Odin login name, not your student number, because your student number isn't in D2L and they're trying to talk to each other. Because of that, and as long as that happens, I should be able to get your clicker scores posted within a couple of days of class. You can check and make sure that your clickers are actually working and that they're getting scored, etc. Which brings us to our first clicker question. So everyone get them out if you have them with you. From a molecular point of view, there's the most diversity in bacteria, oops, we starting? Yes, animals, plants, fungi, or humans.
What frequency?
Double A should be the default, hopefully.
Hold down the power button until the green light blinks and then click double A.
Yeah, so hold down the power button until the green light blinks, click double A if you, to change it. And afterwards, we'll have some time if you want to come down here, and we can make sure that those are set up properly. There's no class after this, which is quite nice.
If you registered with your student ID, will you still get it?
If the question is if you registered your clicker with a student ID, I would try and find you, but it'll take longer to get your scores uploaded. And you can re-register. You can re-register as many times as you like. So once is not too many. Yeah?
Do you know if we were registered in our clicker last term, if we have to do anything?
So the question is, if you register your clicker last term, should you have to do anything? Hopefully not, and that's partly why I'm asking clicker questions today, so we can check and make sure that they get uploaded. Other clicker questions before we count down here. Usually I do 90 seconds for clickers. What do people think? Molecular point of view, there's the most diversity in bacteria. 85% of people think that. Yes, that is definitely true. That's getting back to the tree that I showed at the beginning of the lecture of last year. We looked at the molecular diversity of all cellular organisms. There's far more diversity in the bacteria than all of these other things combined by any stretch of the imagination. So we'll actually select the answer here, close our results. Our next clicker question is, which of the following is the best description of genes that are in a gene family? Hey, maybe we'll have the good answer to this. They're homologs, they're paralogs, they're orthologs, they're paradoxes, they have identical sequences. Oh, by the way, clicker questions. The idea here is really not so that you're getting points and your neighbor's not going to get them. You should be chatting to people about these things. So if you have a question and you think your neighbor knows better, talk to them. If you think your neighbor doesn't know better, then shut up. But still, the idea here is this should be, and hopefully somewhat interactive. We don't have all the chairs and the Poggle and everything else here, but this is sort of my attempt at moving in that direction. So if you have questions about it, please discuss amongst yourselves what you think the answer might be. This, by the way, is also one of those typical obnoxious Stedman questions that what's the best description? Yeah.
We don't have our iClickers. It's been delayed in shipment.
Yeah. Clickers don't count until next week.
Cool. Oh, it's great.
Yes. Yeah, no, so that's, I thought, wasn't that in the first lecture? It should have been. Yeah, so no, clickers don't count until next week. This is basically a way that you can make sure that your clicker is appropriately registered and I can be checking them. So, a little bit more of a difference of opinion here. Homologs, paralogs, orthologs. A couple people like my paradoxes. Good. And identical sequences. Hopefully, and I thought I'd read the description here. We answered the question a little bit earlier. Protein families are all going to be homologs of each other. They could be paralogs. They could be orthologs. And so if you've got a similar, i.e. homologous protein, come from the same source originally in one organism, that can be part of a protein family, or in different organisms, it can be part of a protein family. And so the best description for this would be that they're homologs. So let's get that registered properly. Boom. Okay, so that was our wonderful snowstorm, ice storm. Yes? The gene family, protein family are totally interchangeable, yes. Okay, other questions, comments, worries? Good. Okay, so basically, I always start out my lectures with a little review. Of course, if we've had a lecture beforehand. Try and go over a couple things that I think I did a poor job of explaining or that people seem to be a little bit confused about. So we've already talked now about gene families, hopefully, ad nauseum. A little bit about genomes. I think sort of big take-home message as far as genomes is concerned is that the size of a genome, i.e. the number of nucleotides, the number of genes, has very little to do with what we think about in terms of complexity. We like to think that we're a little bit more complex than amoeba, but amoeba have way bigger genomes than we do. And the central dogma, DNA goes to RNA, goes to protein. There's a little bit of RNA going back to DNA, of course, from reverse transcription. But basically, that's sort of the focus, really, of the whole course, is talking about DNA replication, what do you do with DNA, DNA going to RNA, and then RNA going to protein, and how all of that's regulated. A lot of the best understanding that we have of molecular biology, and particularly replication, transcription, translation, comes out of the study of model organisms, because it's a lot easier to understand what's going on in one organism. You've got multiple researchers working on one organism or one very closely related group of organisms, and there are a lot of experiments you can do on mice or on bacteria that you're not going to be able to do on humans, at least not ethically. So we kill on the order of a couple of billion bacteria on any given day, and nobody complains. If you try and do that with humans, we'd have some issues. So the other thing with model organisms that I wanted to remind you of is that picking model organisms that are as molecularly diverse as possible, so bacteria, archaea, and eukaryotes, finding things that are similar between those is going to tell you something about the real fundamentals of how molecular biology works. And we're mostly going to be talking about those fundamentals. The exciting differences that you see, those are for your 400 level classes, hopefully after this. Any more questions about Lecture 1? Now that we're moving along here. Also, as far as Lecture 1 is concerned, the best way to slow me down is to ask questions. So, talk about chemistry. Again, last time I mentioned, oh yeah, slow down. Yep, good.
Model organisms.
Yes.
Are there new ones proposed often and big fights about that, or do they kind of
So the question is, what happens about model organisms and how often do new model organisms arise? It's mostly organic. It's sort of someone proposes that they have a new model organism, and then that's the one which gets carried forward. Lots of people do really good work on that organism, and then if you've got multiple researchers working on that, then we're going to continue. There's a great movie that just came out called The Fly Lab. Anybody seen that? It's been at OMSI. About Drosophila and the very developments of Drosophila and Drosophila as the genetic material, or Drosophila is using it as a great genetic system. So again, there's some great stuff. I'm trying to push Sulfolobus as being the archaeal model organism, but that's a little bit tougher. So yeah. This question? Yeah.
So with model organisms, I believe that they are also the ones that have been used very notably in the past, like there was a type of zebra something or other that was used extremely well to figure out this revolutionary thing that we didn't know before, and so it's still a model organism even though we aren't doing as much on it now. Is that correct or not?
So the question here is about, to some extent, now how long do people keep using model organisms, and how do you switch from model organisms to non-model organisms? And there's, in fact, a lot of people moving into non-model or novel model organisms, partly because there are a lot more tools that are available for all kinds of organisms. It used to be, and again, getting back to Drosophila, that there were only really good genetic tools for a couple of organisms, including things like Drosophila, because multiple researchers have been working on them for long periods of time. Now there's some pretty general genetic tools. CRISPR-Cas9 is also a nice example of something that's not even really in this textbook in terms of how things are moving rather rapidly. It's how you can make genetic systems out of pretty much any organism that you're interested in. So it depends really on the questions you're trying to answer. Zebrafish, which is the model organism you brought up, in terms of some systems, people are still using zebrafish. It's a wonderful model organism. It's a great genome now, which is complete, well-annotated genome for zebrafish. And so a number of people are still using zebrafish. There's a great lab, in fact, down at the U of O, which is one of the first labs to develop zebrafish as a nice model organism in terms of a good vertebrate model organism that's a little bit easier to work with than mice. So yeah, it varies quite a bit. People are branching out, I think, into many more of the non-model organisms, but there still a lot of work going on in model organisms, a lot of great work going on in model organisms. Okay, so chemistry, as I mentioned last time, this is all the chemistry you need to know for molecular biology. It's not all the chemistry you should know. It's a lot more. But as far as molecular biology is concerned, so we'll talk a little bit about atoms. Mostly as far as molecular biology is concerned, it's all about the weak interactions because these are the weak and particularly reversible interactions that you can have between different molecules, and usually it's going to be macromolecules as soon as we're talking about molecular biology. So it's going to be your DNAs, your RNAs, and your proteins interacting with each other. And the key there is that it's going to be a reversible interaction. So these are van der Waals interactions, hydrogen bonds, and ionic interactions. Absolutely critical as far as biology is concerned is water. Some of you may have been following what NASA has been doing. A lot of my research is actually funded by NASA. One of the things that NASA does is follow the water, because water is absolutely essential for life as we know it. And we'll talk a little bit about the chemical aspects of water, mostly having to do with hydrogen bonds that water forms. Strong interactions, we'll talk a little bit about these, actually much less critical as far as the activity of most molecular biology, but it's forming the macromolecules, which requires lots of strong interactions, particularly covalent bonds that form, and then a lot of modifications that happen too. Particularly proteins are going to be happening due to covalent bonds. And then how do you make these? All has to do with high energy bonds and coupling. Hopefully most of this is review. If this stuff is not review for some of you, you might want to come and talk to me afterwards about whether this is the right place for you to be. Atoms in biology, basically this is great because you only have to care about 4 of them. Hydrogen, carbon, nitrogen, and oxygen. That's by far and away the majority of atoms that we have to deal with. Okay, occasionally we have to deal with phosphorus, sulfur, chlorine, sodium, magnesium, potassium, and calcium. The rest of them, basically, as far as certainly this molecular biology course is concerned, they're not going to concern us at all. And just a quick shout out, however, to the inorganic chemists out there. 4 new elements. Anybody get the 4 new elements? So yeah, so in fact, this whole bottom row here is now complete according to IUPAC as of the end of last year. I was almost a chemist in a previous life. So, a couple things about our chemical bonds. I really hate this terminology here, but it's what's used. This is in fact the older version of the textbook, but the same figure. This is really an ionic interaction. I wouldn't call it an ionic bond. It's when you're sharing electrons. That's where I consider we have a bond, and particularly your covalent bonds. It's when you've got electrons that are being shared between 2 atoms. Here, your ionic interaction is merely really much more of an electrostatic interaction. It's negative and positive charges interacting with each other because one of the electrons has been completely transferred to another atom. What's important, again, as far as molecular biology is concerned, really has to do with how much things are moving around. Just because we are aqueous, most of us is water. All life that we know of is dependent on water. Most of us are at around 30 degrees Celsius, at least inside of us. Average thermal motions bounce things around quite a bit, so there's a certain amount of energy. Non-covalent bonds, these are the ones that are really critical in terms of molecular biology because they're reversible. They come on and off. You need lots of them. On the other hand, making and breaking covalent bonds requires a whole lot more energy. It's a log scale here. It's kind of a back-of-the-envelope kind of calculation. If you've got a covalent bond, it's about 350 kilojoules per mole. Average thermal energy is about 100 times less than that, which means that thermal energy is not going to be breaking covalent bonds. On the other hand, thermal energy is very similar, in some cases even higher than some of these weaker interactions. The van der Waals interactions are extremely weak, so one particular van der Waals interaction is going to get broken off immediately by just the thermal activity of the water, Brownian motion, etc. Hydrogen bonds, not going to do too terribly well. Ionic interactions, and these are the stable ionic interactions in water, are still better, stronger, but you really need a lot of them in order to get these kinds of interactions working properly. So with that, I wanted to show you the video here. There are a number of videos that are connected with the textbook. Some are in the 5th, some are in the 6th edition. This I thought was quite a nice one. Here the commentary is a little weak, so we won't actually listen to it here. But the idea is that we've got a molecule here that's going to be interacting with other molecules. Here, if there are a relatively few number of interactions between these, is your non-covalent, weak interactions that are happening between the two. You're going to have relatively short periods of time that they're interacting with each other. On the other hand, if you have multiple of these non-covalent interactions here running away from us, that's going to be much more stable. They're going to be interacting with each other for a much, much longer period of time, but still dissociating and reassociating. So I think, if I remember correctly here in just a second, they have they're interested with a long time, there it dissociates, but then it'll reassociate again really quite rapidly with another one of these molecules. The other important thing about this is this particular animation is nowhere near as crowded as it should be. Most animations give you a vastly oversimplified version of what's going on inside the cell. The cell is really packed. There's macromolecules, various different RNAs, DNAs, certainly in the nucleus, everywhere if you're a non-nuclear or nucleated organism, proteins. So these things are much, much more closely packed to each other. It's really hard to visualize under these conditions. Yeah, the question?
What was the takeaway from that movie?
Takeaway?
Yeah.
That Multiple interactions are important for getting any kind of interactions between these macromolecules. Second thing is it's not permanent. They are going to come off and reassociate, associate, and disassociate from each other. It's the number of those interactions which is the really important aspect of it. Okay, I keep beating on water, and we'll continue to beat on water for the rest of the term, probably, because life as we know it really depends on water. Why is that so important? It really has to do with the polar nature of water. And I mentioned, again, I hate to keep referring back to the previous lecture that some of you I know haven't watched yet. There's really two kinds of polarity I'm going to talk about in this class. There's polar molecules, which are going to be things like water, that'll have slight differences in charges depending on either end of the molecule. I'll also talk about molecular biology polarity, and that's going to be having a start and a stop, and that's particularly important for nucleic acids. There's going to be a 5' end and there's going to be a 3' end. And that's true for all nucleic acids. For all proteins, there's going to be an N-terminus and a C-terminus. So when I talk about molecular biology polarity, it's going from a 5' to a 3', or from an N-terminus to a C-terminus. If I talk about something which is polar, that's going to be something like water, which has this separation of charge. So I will keep beating on particularly the molecular biology polarity, because it's really important that you have a start and a stop, and it's a linear progression between those two. It's one of the things that makes polysaccharides really hard, because they're not nice and linear. Okay, this makes sense? Yes? No? I haven't had enough coffee yet this morning. So polarity of water, again, is because it doesn't have this nice symmetrical molecule. It's got a very definite asymmetry to it, where you have slightly positively charged end of the molecule and a slightly negatively charged end of the molecule. And basically what that means is that hydrogen atom and its associated electron are going to be able to associate with other slightly negatively, or if you think about the hydrogen ion, slightly positively charged, will be repelling these particular interactions. So that forms the hydrogen bonds. Again, you're not sharing electrons here, so I don't like the terminology hydrogen bonds, but everybody uses them. So the hydrogen bond is just sharing your hydrogen, and particularly the electrons of that hydrogen, between two other atoms. In this case, nitrogen and oxygen, but you can also very easily have hydrogen bonds between two different oxygen atoms. What particular example? There are a whole bunch of those. Alcohols. And what about water? What about that molecule I just talked about? So water is wonderful at forming hydrogen bonds. It's probably why it's so absolutely critical for life as we know it, because they're interacting with each other here. But we'll also see hydrogen bonds that form, particularly important for nucleic acid structure, between NHs and Os. So amide and then oxygen atoms, which are involved there. A couple of important aspects about these shared hydrogens, or the hydrogen bonds. Again, they're about 100-fold less than general thermal energy at 37 degrees Celsius. Very important is that these are a directional kind of interaction. If you don't have the hydrogen lined up appropriately between the different atoms, you're not going to have that 4 kilojoules per mole. I'm trying to use kilojoules. Kilocalories is a thing that dies very hard. But 4 kilojoules per mole interaction between those things, but only if they're lined up properly with each other. If they're not lined up properly, you're not going to have that interaction. So it's a very specific, and this is great in terms of thinking about macromolecular interactions. They've got to line up perfectly with each other. Otherwise, you're not going to have those kinds of good interactions. On the other hand, van der Waals interactions are just getting close to each other. There's no directionality to van der Waals interactions at all. Some people talk about van der Waals bonds. I don't like that. I much prefer interactions, and more people use interactions for this too. It's because there's a big difference depending on the distance between the atoms. So at about 3.2, about 3.5 angstroms away from each other, you have an attractive force due to van der Waals interactions. But as soon as you get shorter than that, actually that starts to be a repulsive force. So it really holds these two atoms at a very specific distance relative to each other. And very often, and we'll see lots of models of this, you see the ball and stick models, or particularly what people call space-filling models of macromolecules, or for that matter, just even molecules. Those are going to have the van der Waals radii on those, and that radius is going to be exactly where you have this optimal attraction. As soon as you get lower than that, they start to get closer together, they're going to be repulsed from each other. So van der Waals interactions are both positive and negative, and again, relatively short relative to each other, and then you get fewer and fewer interactions very, very rapidly as soon as you get further away. So hydrophobic interactions. A lot of people talk about hydrophobic bond. Anybody heard about a hydrophobic bond? Doesn't exist. So all that hydrophobic interactions are is really hydrogen bonds that are forming between water and not forming with whatever the hydrophobic molecule happens to be. So if you look here in this image, we've got lots of hydrogen bonds, these little yellow lines here, between the water molecules. Certain number of hydrogen bonds here, you don't need to count them up. There are a lot more hydrogen bonds now when these two molecules are associated with each other. But all that you're doing here is maximizing the hydrogen bonds that you have between the water molecules. It's not these two hydrophobic molecules interacting with each other. It's the water interacting with itself. It's not a very strong interaction, hydrogen bonds, but there's a heck of a lot of water there. And maximizing those interactions is going to be pushing these otherwise hydrophobic, i.e. not interacting, not making hydrogen bonds with water, towards each other. And so it's that pushing interaction which is there. So macromolecules, all about lots of weak interactions. We already looked at the movie. Basically, again, it's that the take-home message there is that we've got multiple weak interactions, that you have transient interactions between two macromolecules, or sometimes a macromolecule and a much smaller molecule. And that's just going to be longer as long as you have more of these interactions with each other. So questions about these various different bonds. We'll move on to chemistry. Okay, the chemistry that you need to know for this class is very basic. I didn't mean that pun. Basic and acidic. But as far as we're concerned, acids are molecules that can give up hydrogen ions, and bases are those things that can pick up hydrogen ions from solution. It's almost always going to be in an aqueous solution, although you can actually have these interactions between things that are not water. But for the vast majority of these kinds of interactions, it's going to be interactions that you have with water in any particular case. So that's all that I wanted to mention here. What are the main molecules? We're going to be talking about adenosine triphosphate. Hopefully everyone knows ATP. Yes, good. If you don't know ATP, probably the wrong place for you to be. Adenosine triphosphate is a nucleotide. serves as a building block for the nucleic acids, which we're going to talk about almost all of next week, and to some extent on Friday as well. These are the also, again, monomers which go together as nucleic acids, but ATP is also the energy source for the cell because it has all of these covalent bonds, particularly the phosphoanhydride bonds here, that can be broken, and that energy can then be used for other aspects inside the cell. Proteins, we'll talk a lot more about proteins, well, at this rate probably more on Friday than we do today. Sugars, which are made up of these individual sugar residues. We're not going to talk any more about lipids than just look at them right here. Lipids are very important, but not as far as this molecular biology is concerned. What are cells made of? The vast majority of cells is water. That's true for a particular bacterial cell, also true for eukaryotic cells, also true for plant cells. The vast majority of the what makes up the volume of a cell is going to be water. And again, that's because of all these wonderful hydrogen bond interactions that water can form and allows all of these other interactions really to take place. Of what's left, most of the remainder is protein. There's a lot of RNA inside cells. It's going to be messenger RNA to some extent, but that's actually a relatively small amount of the RNA which is there. It's mostly ribosomes and transfer RNAs, which are present inside the cells. And now we know more and more about some of the regulatory RNAs, but those are relatively small amounts. So mostly water. What's not water, about half of that's protein, quite a bit's RNA, and then the other stuff is really pretty minimal. So let's talk about some of the chemical reactions that take place in this aqueous solution. So all of biochemistry is aqueous interactions. And for the most part, all the reactions we're going to be talking about have to do with either taking away water or putting in water into your molecule. So condensation reactions, hydrolysis reactions, removing water is a highly energetically unfavorable reaction. It gives you, in most cases, a covalent bond. We'll talk about how you can do that in the next couple of minutes. On the other hand, hydrolysis is a highly favorable interaction that happens, partly because you have a huge amount of water around. Remember, 70%, which is there, molecularly very high concentrations. One reason why it's energetically favorable. The other reason why it's energetically favorable is this is raising entropy. So you're getting more disorder in your system. So those are the two reasons here. But as hopefully we all know, we need all these macromolecules, and they all come from these small molecule subunits. So there's got to be a lot of condensation reactions going on, which is exactly how you go from these individual nucleotides to polynucleotides. And you have to avoid hydrolysis. How do we get that to happen? Well, it's because we have coupling of reactions. So you have an unfavorable reaction and a favorable reaction, just from an energetic point of view. And then you take the energy you get from a favorable reaction and use it to allow you to have unfavorable reactions take place. And so that's just shown here, where you have the free energy, and we'll talk a little bit more about free energy in just a second. Hopefully it's all review for you. Free energy of A is lower than B, so this is going to be an unfavorable reaction, but B to C is a favorable reaction, so if you have B to C, you can get A moving to B. Same thing is true of C to D, and then D to E. So A to E by itself is what we'll call a pathway, and be a lot more of this. Anybody taking biochemistry, by the way? Okay, so this is all a review for you. You can correct me when I screw up. So all these pathways, are going to be favorable. You add up all of the free energies of all the reactions that you're looking at, but most of the reactions we're going to be talking about, which are our condensation reactions, making macromolecules, making covalent bonds, those are going to be unfavorable. So basically, you take high energy food molecules, if you want to think about it from a purely anthropomorphic sense, break them down, you lose heat in this process, so it's the entropy increasing. Heat is just motion. And then harness some of that in order to make these molecules, the macromolecules which are there. I love this image, actually one of my favorite textbook images of all time. So this is the high energy state. This is the low energy state. Spontaneously, particularly if you have, as I like to call them, viral vectors in your vicinity, it will go from here to here, and then you must input energy to reverse this entropy to go back over to this state. The way that happens on our planet is mostly, although there's, anyone had microbiology? Some of you? Yeah, so you know this is oversimplification because there's also nice chemosynthesis that happens. But as far as we're concerned, most of the energy that we use on this planet comes from sunlight. And that sunlight, through photosynthesis, basically takes water and CO2, makes it into macromolecules, and then from those building blocks, I should say, and those building blocks are then used to make macromolecules. That process, again, it's the energy of sunlight that then gets broken down, again, through breaking bonds, so you can then form bonds, and that gives you the macromolecules, which are what really is forming life. Again, it's an oversimplified process here. Photosynthesis is going to take carbon dioxide in the atmosphere, convert it into sugars. That then will get taken up by us and all the heterotrophs, which will then respire, which gives off the CO2, consumes the oxygen, and we go around and around and around. Of course, as I alluded to here, many microbes are chemosynthetic, and so they actually use different energy sources than the sun. But we, for the most part, are very much dependent on that big yellow thing you see occasionally here from time to time, being that we're in Portland. Briefly wanted to talk about oxidation and reduction. This is, again, should be very much general chemistry. I love the Leo and GER. So what's Leo stand for? Loss of an electron is oxidation, and gain of an electron is reduction. So that's the way I like to remember these things. In the textbook, they also mention hydrogenation or dehydrogenation. So just looking over here at the example from going from methane to carbon dioxide. Carbon dioxide here is the most oxidized. Methane is the most reduced. And that's the loss of the electrons is oxidation. So we're losing the electrons here that are associated with the hydrogens, ending up all the way down here at CO2. The gain of electrons is reduction. You can also look at this in terms of losing the hydrogens, which is an oxidation, gaining hydrogens, which is reduction. And there's almost always going to be exchanges here, oxidation and reduction reactions. So you'll oxidize one molecule in the process, reduce another molecule. And so that's how you're maintaining the energy in this process. You can also think of a covalent bond, and particularly a polar covalent bond, again in the chemical polar sense. What's our favorite polar molecule? Water. Where you now have a partial loss of electron here on this side, so that would be partially oxidized, and partial gain of electron over here, and so this is going to be reduced. But again, it's this oxidation reduction reactions which are absolutely critical for most of those early steps. You know, just if you think about photosynthesis, what does photosynthesis do? It pumps electrons into things. And so it gives you then reducing power that you can then use for oxidation reactions. And so that's the process. What do these reactions look like? We already looked at the change in free energy that you get between one molecule and a different molecule. This is the difference in free energy would here between B and C, but for the vast majority of molecules that we're going to be talking about for this class, Y doesn't spontaneously become X. It has to have some input of energy before it can become X. And that input of energy could be just thermal interactions. So in that video, you saw the molecules bumping into each other. That's going to be energy. That could be sufficient energy to get across here, but usually it's not. And if it were enough, then we probably would never see any of molecule Y here and only ever see molecule X. So what you have in most biological systems is some way of stimulating this reaction. So from Y going to X. And that stimulation of the reaction has to do with the rate of the reaction. And what does that are enzymes. Most enzymes that we know of are proteins, although there are more and more enzymes that people are discovering that RNA can actually do the same thing. But most of the enzymes that we know of today, these are all using protein enzymes. All the enzymes do is lower this activation energy barrier. And so basically you can get a more rapid conversion between these two different states. But you don't change the actual equilibrium, and this lowering of this activation energy means that it's actually also easier to go from X back to Y. So enzymes don't change the equilibrium or direction of reactions. They just speed them up. So what do you get when you've done this? Again, hopefully this is kind of beating the dead horse. Reactions that are unfavorable have to be coupled to reactions that are favorable. If you have an enzyme-catalyzed reaction, you don't need as much of that favorable reaction that you have to couple to the unfavorable reaction. But still, you've got differences in the energy of these molecules. You've got to get them from one to the other somehow. How do you do that? That's through these changes in energy. You have the reaction of C to D, which is then coupled to the reaction of X to Y. C to D is a highly favorable reaction. X to Y is an unfavorable reaction. The magnitude of this favorable reaction has to be larger than that of the unfavorable reaction. Otherwise, it's not going to happen. So you have to have lots of input, and that's why we need to eat, why we need sunshine. You have to have an input of energy into the system in order to get us and life to actually happen. Let's talk a little bit about chemical equilibria. The chemical equilibria that are going to be most important for this class are going to be interactions between various different macromolecules or macromolecules and their so-called ligands. But basically all that a chemical equilibrium is at a given concentration of your reactants and products, you're going to have a certain amount which are not reacted with each other, a certain amount that are reacted with each other, and that's going to go back and forth. And at equilibrium, you're going to have a stable concentration of your product and a stable concentration of your reactants. That's best described by equilibrium constants, which are just the ratio of the two different rates. So the rate of formation divided by the rate of dissociation in this particular reaction. You can also write equilibrium constants as the product of the concentration of your reactants. So here, R will be your reactants. Again, and products, in this particular case, we only have one product, and that will give you your equilibrium constant. Why do we care so much about equilibrium constants? Because equilibrium constants tell us something about the change in energy. So the equilibrium constant under standard conditions, none of us live under standard conditions, but at least this is a way for comparing reactions to each other, is 10 to the minus delta G divided by 2.6 times the gas constant and the temperature. But because of this relationship between delta G and the equilibrium constant, if you know the equilibrium constant, you can calculate delta G. You can get delta G if you know the concentration of your products and reactants. If you have standard concentrations, i.e. 1 mole of each of your products and reactants, then you get delta G0, and that gives you the connection between these two. So you can figure out the energy of your particular reaction if you know the equilibrium constant and the concentration of your products and reactants. Why do we care? Well, again, it's mostly because of interactions between different molecules. And that's really all that molecular biology is about. It's just interactions between different molecules. Yeah?
What we have to memorize those equations?
Would you need to memorize these equations? The answer is no. If I gave you an equation like this on an exam, as I give you a question like this on an exam, I would give you the equation. You should not need calculators for any of your exams. If you can't do basic multiplication, 1 or 2 digits, we can talk about that too. But you really should not need calculators. And I'll give you these kinds of formulae if you need them. You may need to memorize these formulae for other classes, but not in this one. So hopefully biochemistry, they force you to memorize these things. So I will let you know if there are things that I expect you to know in terms of formulae, etc. Yeah?
So we should just be knowledgeable about how to manipulate the equation.
Yeah, and know that if you know your delta G, you can get your equilibrium constant, and equilibrium constant, you go back to your delta G. Yeah, and that's basically what this is about, is using that information to get to this particular, these kinds of interactions here. So equilibrium constant here, we're talking about equilibrium between interacting and non-interacting. And this is what people will call dissociation constants. So your dissociation constant is just between your protein bound to a ligand, or two different macromolecules associated with each other. So the product that you're talking about here is ligand plus protein. And then your reactants are ligand and protein. And so the dissociation constant is just going to be these relative concentrations, or the rate of interactions versus non-interactions. And we can do the math later on this. But if you look at the dissociation constant, will tell you the concentration of your partner, in this case the ligand, when half of your protein is bound to it. So this is really important if you think about interactions that are happening between, say, DNA and DNA binding proteins, between an enzyme and its substrate. If you have a very low KD, that tells you that you have very small amounts of unbound ligand. If you have a very, very high KD, That means you've got a lot of unbound ligand around. And so that will tell you how much interactions you have with your DNA molecule or with your enzyme at any particular time. Yeah?
What's the sub-name mean?
This is the dissociation constant, so Kd as a dissociation constant. All it is looking at the equilibrium between bound and unbound. So it's just a slightly different way of looking at things. But this is going to be a very important aspect of things. We go to 11:20, correct?
Correct.
Good, okay. So we'd have 10 more minutes here.
On the last slide?
Pardon?
On the last
Here, yeah.
I don't think it's what you just asked.
Okay, sure.
I'm sorry. What is the little m, large M, and then N, large M, KD weak, KD strong?
Oh, okay, yes. So here, down at the bottom. So the question is here, what does he mean by all these funky stuff down here at the bottom? Thank you for reminding me about this. So I just mentioned that if you have very low KDs, you have very strong interaction between things because you have very little of your unbound ligand, whereas if you have a relatively high KD, you're going to have a pretty high amount of not bound. So this is millimolar dissociation constants, which is people think about as weak kinds of interactions on a molecular biology scale, whereas a nanomolar interaction is going to be a rather strong interaction between the two. So that's what this means down here at the bottom. That's if people will talk about strong interactions or weak interactions between, say, a protein and a ligand or two different macromolecules with each other. If you're talking about millimolar dissociation constants, that's going to be a weak interaction. Nanomolar is going to be a very strong interaction. Okay, so let's talk a little bit about some of the reactions that have to take place. I've been again beating around the bush on a lot of these things. Mostly we care about polymerization. We care about going from monomers to multimers. We care about going from nucleotides to RNA, nucleotides to DNA. We care about going from amino acids to proteins. So how does that happen? You have to form covalent bonds. Covalent bonds are pretty high energy. We know that what we're happening here is this is a condensation reaction. We are removing water. Again, this is going to be challenging energetically. How do you do this? Well, it's basically through our good friend ATP, or for that matter, a number of high energy intermediates. And so these are the high energy covalent bonds that when you break them, you get a lot of energy out of that. And as long as you can store that energy somehow, that will allow you to make these unfavorable reactions. And again, for the vast majority of things in molecular biology, that's going to be a polymerization reaction taking place. Here, it's actually not a polymerization reaction, but it's an example from the textbook on how this happens. You have a glutamic acid. This is one of the amino acids necessary for proteins. You need a glutamine. How do you get there? You hydrolyze ATP. So you lose one of the phosphates from the ATP molecule. That phosphate gets transferred to this OH on the glutamic acid. And then this reaction of condensation, the addition of ammonia, is a reaction, and I don't like that it's higher and lower energy here. This is actually a higher energy molecule than that. But you use this high energy intermediate with these phosphoanhydride bonds in order to make these otherwise impossible, at least from an energetic point of view, reactions take place. So let's look at polymerization. Again, this is lots of covalent bonds that have to form. You need lots of high energy bonds in order to make these multiple covalent bonds. The other thing with polymerization, as I mentioned before, all these chemical reactions are reversible. You don't want, once you've spent all this energy to make a macromolecule, you don't want to be reversing that. So you want to have ways of making polymerization be as irreversible as possible. The way that happens, which seem to be losing the images for here, which is interesting. They just showed up on the screen before. And I would scribble all over the board here, but it would be completely left over. But basically what happens in terms of polymerization is not at all, not that different from what happens here. So for your glutamic acid, you have hydrolysis now of the individual nucleotide. The individual nucleotide will get to this high energy intermediate. What happens, though, instead of going from ATP to ADP, you have ATP that goes to AMP, and then you end up with pyrophosphate. Pyrophosphate is two phosphate molecules hooked together with a phosphoanhydride bond. That bond then also gets hydrolyzed, and then you have AMP plus phosphate plus phosphate. That is basically now an irreversible reaction. You're not going to hook up the AMP to each of these individual phosphates, and so the irreversibility that you get from polymerization that should be on this slide, and I have no idea why it disappeared. So it's in the textbook, and I could bring it up, but in the last 5 minutes it's hardly worthwhile doing that. Here you have pyrophosphate that forms, the energy you get from hydrolysis of that first bond. The energy that you get from the hydrolysis of the second bond between those two phosphates is actually not really used for your polymerization reaction. It is, however, the critical part of this irreversibility. You're not going to be adding back a pyrophosphate because it's been hydrolyzed. And so that's how you get the nucleic acid polymerization, which is happening and will be shown on this slide. ATP goes to AMP plus pyrophosphate. Pyrophosphate goes to two phosphates. And that gives you your irreversibility of this process. You have an activated intermediate, which I'll bring up and we'll have next time, that we can take a look at and see those high energy intermediates that you have during the nucleic acid polymerization reaction. How do you do this? It's through the activity of an enzyme, surprise, surprise, that's the DNA polymerase, which we will talk a whole bunch more about in a couple of weeks. So what did we talk about today? Talked about atoms. Again, fortunately, we only really need about 4 of them: hydrogen, oxygen, carbon, and nitrogen. Exactly. Slightly less important are phosphorus, sulfur, and then some of the ions. Weak interactions, lots of weak interactions that you have to have in terms of getting stable interactions. Got lots of them. You're going to have dissociation constants, which are going to be high or low. Lots of interactions. So you'll have low dissociation constants. That's a strong interaction. So lots of interactions there. Water is critical because it's got all of these hydrogen bonds that it can form. And I did want to mention in the textbook, and this is in both versions of the textbook, if you need a review, there are these panels, which are blue pages here in Chapter 2, which really do a nice job of going over some of the chemistry, etc., which is there. And I'm pretty sure they're in the 5th edition as well as in the 6th edition as well. So we'll talk about proteins and some nucleic acids on Friday.